This video will explore Chapter 3, Biological Molecules, the Carbon Compounds of Life. Remember, Chapter 1, Chapter 2, and Chapter 3 lecture notes will be covered on Exam 1. Use the study guide on Blackboard to prepare for Exam 1. By the end of this video, and subsequent review of Chapter 3, you should be able to discuss the learning objectives, using information from Chapter 3. Along with hydrogen, oxygen, and nitrogen, carbon makes up more than 96% of the weight of biological organisms. There are four major biomolecules important to living organisms: carbohydrates, lipids, proteins, and nucleic acids. These organic, or carbon and hydrogen-containing molecules, primarily use covalent bonding strategies, and will be described in this chapter. Life on Earth involves both organic and inorganic molecules. You will recall that carbon has the capacity to form a total of 4 bonds, given the fact that it only has 4 valence electrons. This allows carbon to form a wide variety of biological molecules with many shapes and sizes. This figure illustrates the diversity of hydrocarbons in nature. When considering the diversity of hydrocarbons, keep in mind that molecules primarily differ in structure and function because of their functional groups. Functional groups are a collection of atoms in an organic molecule that are responsible for its observed chemical properties. As their name implies, a chemical functional group gives a molecule its functions in a biological setting. The most important functional groups in biology are the hydroxyl, carbonyl, carboxyl, amino, phosphate, and sulfhydryl groups. Proteins, nucleic acids, carbohydrates, and lipids are made using a process known as dehydration synthesis or condensation reactions, and degraded using a process known as hydrolysis. In dehydration synthesis, monomers are covalently bonded to form polymers with the subsequent release of a water molecule. In hydrolytic reactions, polymers are broken down to monomers with the addition of a water molecule. Look at the graphics on this slide that represent dehydration synthesis and a hydrolysis reaction. In a dehydration synthesis, molecules are joined. In a hydrolytic reaction, molecules are broken into simpler parts. Again, in a dehydration synthesis, water is lost or removed. In a hydrolysis reaction, water is added to disrupt covalent bonds. Take the time to review this sample Exam 1 question. If you have any questions about this slide, contact Dr. Flowers via email. Chapter 3 focuses on the 4 major macromolecules of living organisms. The 4 major macromolecules are proteins, nucleic acids, carbohydrates, and lipids. The terms polymer and monomer are general terms that can be used to describe biological molecules. A monomer is a molecule consisting of one part, while a polymer is a molecule consisting of many parts. Monomers combine to form polymers. For the exam, know the difference between a monomer and a polymer. For example, an amino acid is a monomer, while a protein is a polymer consisting of several to many amino acids. Also, for the exam, understand the similarities, differences, and functions of each of the 4 macromolecules. Typically, a polymer greater than or equal to 1000 Daltons is considered a macromolecule. The word macro means big, so a macromolecule is a big molecule. Carbohydrates are essentially sugars that provide 2 basic functions for living systems. They provide the building blocks for macromolecules and serve as major energy storage compounds. Carbohydrates are also called saccharides. Depending on how many monomeric subunits exist, a molecule can be a monosaccharide, disaccharide, or polysaccharide. A monosaccharide only contains 1 sugar. A disaccharide contains 2 sugars. A polysaccharide contains many sugar monomers. Carbohydrates typically have the same ratio of C, that is, 1, 2, 1. For every one carbon molecule, you will find 2 hydrogen atoms, and 1 oxygen atom. One of the most famous monosaccharides in a biological context is glucose. Later on, in the semester, we will discuss how glucose is used by humans to produce energy in the form of ATP that is used to power our lives. Ring formation is possible when a monosaccharide contains at least 5 carbons. A disaccharide is a sugar consisting of 2 simple sugars. A polysaccharide is a sugar consisting of 8 or more monosaccharides joined via dehydration synthesis. Some examples of polysaccharides include starch, cellulose, chitin, and glycogen. Take the time to review this sample exam 1 question. If you have any questions about this slide, contact Dr. Flowers via email. Lipids are non-water soluble or nonpolar organic molecules. Lipids have several functions in living entities, such as energy storage, protective shielding, biological membrane composition, and in the development of hormones. Since lipids are nonpolar, they do not associate with water molecules, and are sequestered in an aqueous environment. In the case of the phospholipid, shown on this slide, you have a molecule with a polar, hydrophilic, or water-loving head and a nonpolar, hydrophobic, or water-hating tail. The polar heads orient themselves toward water molecules, while the nonpolar tails orient themselves away from water during the formation of a biological membrane. Major examples of lipids include oils, fats, triglycerides, waxes, phospholipids, and steroids. A triglyceride is a simple lipid consisting of glycerol and 3 fatty acids. Fatty acids contain a hydrocarbon, linked to a carboxyl group, and are important in lipid architecture and function. Major examples of lipids include oils, fats, triglycerides, waxes, phospholipids, and steroids. A triglyceride is a simple lipid consisting of glycerol and 3 fatty acids. Fatty acids contain a hydrocarbon, linked to a carboxyl group, and are important in lipid architecture and function. In terms of energy storage, triglycerides are more effective energy storage molecules compared to carbohydrates. Waxes are a type of lipid molecule attached to other chemicals such as alcohols and serve as structural and protective barriers in nature. As I said before, phospholipids are lipid molecules that contain a phosphate group. Phospholipid molecules form a lipid bilayer in the presence of water. The lipid bilayer is also called the plasma membrane, biological membrane, cytoplasmic membrane, or cell membrane. Again, the polar heads point toward water, and the nonpolar tails point toward each other in a biological membrane. Steroids are a specific group of lipids, including cholesterol and hormones. They are different from other lipid molecules, in that they are ring structures. A very important sterol, cholesterol, is shown on this slide, and is prevalent in eukaryotic cell membranes. 2 major steroid hormones are estradiol and testosterone. Estradiol is secreted by the ovaries, and is important for women and female development. Testosterone is secreted by the testes, and is important for men and male development. Proteins are large macromolecules made up of amino acids connected via peptide bonds. Amino acids have a very similar atom configuration. They contain an amino group, carboxyl group, and a variable side chain called an R group. The image on the slide shows you the basic structure of almost all amino acids. Proteins play a role in numerous biological functions, as highlighted here in yellow. Proteins determine all of your physical and behavioral traits. Proteins determine your height, weight, eye color, hair color, and many other important traits humans need for survival. Proteins are also called polypeptides. Twenty amino acids are used to make up the majority of proteins in the human body. Again, most amino acids contain a central or alpha carbon atom that is connected to an amino group, carboxyl group, side chain, and a hydrogen atom. The major exception to this arrangement is the amino acid proline, which contains a ring structure. While there are 20 different amino acids, there are 4 types of amino acids, depending on the nature of the R group, or side chain. The 4 types of amino acids are either polar, nonpolar, acidic, or basic. Peptide bonds are used to link amino acids together to form proteins. Peptide bonds are bonds joining the amino group of one amino acid to the carboxyl group of a second amino acid. Proteins are characterized by having 4 organizational levels: primary, secondary, tertiary, and quaternary. Primary structure is the first level of protein structure. Primary structure consists of the specific sequence of amino acids making up a protein. The amino acid sequence is important for protein function. Changing one amino acid in a protein could alter protein function. Secondary structure is the second level of protein structure. Secondary structure consists of alpha helices and beta pleated sheets, as well as the regular patterns of coils or folds of a polypeptide chain. Alpha helical structures are rod-like structures. Beta pleated sheets resemble paper accordions. Tertiary structure is the third level of protein structure. Tertiary structure refers to the three-dimensional shape of a protein. Tertiary structure is the level of protein structure where protein function is expressed. The collection of polar and nonpolar amino acids determine the solubility and functionality of proteins. Take the time to review this sample exam 1 question. If you have any questions about this slide, contact Dr. Flowers via email. The shape of a protein is paramount for the protein's function. Proteins that lose their specific shape, due to unfolding, are inactive or non-functional. Denaturation is the term used to describe the unfolding of a protein. The image in this slide provides an excellent view of protein denaturation. Denaturation is the change in the molecular structure of a protein, usually making it non-functional. A protein's shape is essential to its function. After translation, proteins must be folded to their precise specifications. Chaperonins are used in the cell to promote protein folding. Essentially, chaperonins are a family of proteins within which protein folding takes place. As you can see from the figure on this slide, in step 1, an unfolded protein enters the chaperonin, and leaves the chaperonin fully folded. Misfolded proteins are inactive, while fully folded proteins are active. Quaternary structure is the fourth level of protein structure. Quaternary structure is formed from the association of 2 or more polypeptide subunits, or 2 or more protein subunits. The next 2 slides demonstrate the 4 levels of protein structure, from a visual perspective. Tertiary proteins and quaternary proteins contain specialized regions within the protein that allows the protein to perform specific functions. Moreover, domains are a folded three-dimensional region of a protein that forms the basic unit of tertiary structure. Take the time to review this sample exam 1 question. If you have any questions about this slide, contact Dr. Flowers via email. Nucleic acids are polymers consisting of nucleotide monomers. The 2 types of nucleic acids are DNA and RNA. Nucleotides consist of 3 components. Nucleotides are made up of nitrogenous bases, a 5-carbon sugar, and a phosphate group. There are 2 types of nitrogenous bases, pyrimidines and purines. Pyrimidines are one of 2 families of nitrogenous bases, found in nucleotides. Pyrimidines consist of uracil, thymine, and cytosine. Purines are one of 2 families of nitrogenous bases, found in nucleotides. Purines consist of adenine and guanine. In terms of the 5-carbon sugar in nucleotides, DNA contains deoxyribose, and RNA contains ribose. This figure shows the chemical structure of nitrogenous bases. Nucleosides are compounds consisting of a purine or pyrimidine base, and a pentose sugar. This figure shows that DNA is a polymer, made up of 2 polynucleotide strands. DNA is commonly referred to as a double helix, based on the way the 2 nucleotide strands interact. In DNA, adenine pairs with thymine, and cytosine pairs with guanine. In RNA, adenine pairs with uracil, and cytosine pairs with guanine. While DNA is a double-stranded molecule, RNA is a single-stranded molecule. RNA is produced as a result of transcription. Complementary base pairing refers to the fact that, in DNA, adenine pairs with thymine, and guanine pairs with cytosine. Phosphodiester bonds are found in DNA and RNA. Phosphodiester bonds are covalent bonds formed between the 5-phosphate of one nucleotide and the 3-hydroxyl of another. The next few slides show the differences between DNA and RNA. The next few slides show the differences between carbohydrates, lipids, proteins, and nucleic acids. Take the time to review this sample exam 1 question. If you have any questions about this slide, contact Dr. Flowers via email.